Welcome. In this session, I'm going to discuss about regulation of gene expression. So this regulation of gene expression is a broad term, right? But we will restrict it only to synthesis of a polypeptide chain. So in earlier sessions, I told you that the DNA through the process of transcription, so it forms mRNA, and this mRNA will be undergoing translation, which results in the formation of a protein. So that results in the formation of protein. So this what I call it gene expression. It's called gene expression. So I can say that the process by which, so in case of gene expression, how I can define this gene expression? The process by which genetic information, by which genetic information, so that is whatever that information which is present in DNA will be copied to mRNA, and that will be translated to form protein. So the process by which genetic information from DNA is carried to RNA. So from DNA, it is carried to RNA and translated into a protein. Translated into a protein. Right. So that's what I call it gene expression. Okay. Now let's see in case of DNA molecule. Now we know that there are several genes. Okay, so there is a DNA molecule, and you can see from this point to this point, say there is gene A, and then so there is gap here, and somewhere here is again gene B. Of course, gap means here there will be nitrogen bases, but they are not coding for right any protein or polypeptide that I have left it. So then later here, say gene C. Like this, you will find several genes on the DNA molecule. Now, right at a particular time, a gene is required. Its product is required. So of course, this gets expressed and results in the formation of product, and that product may be enzyme, okay, protein, or it may be right hormone, etc. So in the form, the product can be enzyme, okay, or any other protein, or maybe hormone, some hormones that are proteins, right? So here same that will be in the case of gene B also and gene C also. So what we mean by product, so this is what it can be: enzyme, it can be protein, or it can be, okay, hormone that is a right made up of protein. Now at a particular time, so this product of gene A is required, whereas the products from the gene B, the products from the gene B is not required, and also the product from the gene C is not required. So then what happens? So I'm telling at a given time, so at a given time, right? So this gene B and gene C will not be transcribed, but gene A will get transcribed and translated to form, okay, any of that product. So that results in the formation of the product, right? Now after some time, Now the product of the gene A is not required. So hence this will get switched off, and now say that gene C product is required. Now this one gets transcribed and translated and finally results in the formation of product. So what I mean is that always all the genes on the DNA will not be in switched on condition. Means all of them will not be simultaneously getting transcribed and translated and produce the product, right? It all depends on the requirement of the cell, right? Now basing on it, some genes will be active or switched on, and some genes will be inactive or switched off. So at a given time, so in some cases say gene A and gene C, now they are required, but gene B is not required, so hence it becomes inactive, the others will be inactive. So this is all to conserve energy. So here, if there is a need, then the product will be produced. If there is no need, then the product will not be produced. So that's, let's see, by taking an example in this case, say suppose I'm going to put a E. coli bacterium. So when I'm going to put this E. coli bacterium into a medium containing lactose. So when I put this E. coli bacterium into a medium containing lactose, it's going to synthesize enzymes that will metabolize the lactose. Okay, so when in the medium, the E. coli bacteria will produce enzymes that metabolize lactose, right? Now if I am going to remove this E. coli bacterium out of the lactose, so when I remove the E. coli bacterium out of the lactose, there is no need, because the surrounding environment doesn't have the lactose and there is no need to synthesize enzymes, and even though if it synthesizes, it is waste, and waste of energy also, right? So E. coli bacterium, when placed in a medium containing lactose, then only it is going to produce enzymes that will metabolize the lactose. And out of the E. coli medium, it is not going to synthesize those enzymes. Right? It means that when E. coli bacterium is outside the lactose medium, the genes are switched off. And when in the lactose medium, the genes will get switched on. So that is what I mentioned here, that if there is a requirement of that particular product of a gene, then the gene will get switched on or becomes active, gets transcribed, translated, and then finally results in the formation of product, right? So now how these genes get switched on and switched off, and how they come to know that when they have to become active or inactive, so all that we will study by taking an example. So that is Lac Operon. So we'll consider Lac Operon, right? And of course, this Lac Operon concept was proposed by two persons. So it was proposed by two persons. One is Francois Jacob. So this person is Gentis, and another one called Jacques Monod. and this person is a biochemist, right? So these two persons proposed Lac Operon concept, right, in somewhere 1961. So that actually helps us to understand that when a gene gets inactive and when it gets active, right, depending on the requirement. Okay, so now, as I told you, that the expression of gene will be in terms of synthesis of a polypeptide. Now this expression, that is gene expression, can be, right, regulated at several you know uh, levels. Gene expression can be regulated at several levels in eukaryotes. Okay, why? Because in eukaryotes there is compartmentalization. Whatever that RNA molecules that are going to synthesize, they need to first get synthesized inside the nucleus, and thereafter there is a processing of the RNA molecule. So the processing means introns need to be removed, exons will be joined, and there will be 5 and capping, then third and tailing. Okay, so all these, what I call it, processing of RNA molecule. So such processing of RNA molecule we don't find in case of prokaryotes, right? So there are no introns and no exons. So that what we see, the split gene arrangement we don't find in case of prokaryotic cells. We see only in case of eukaryotic cells. So no RNA processing in prokaryotes, and that RNA processing we'll see only in case of eukaryotic cells. And this gene, whatever the genes in the eukaryotes, that can be regulated uh, at several levels in. Okay, so now let's look at here. So at what levels it can be regulated? So first one, we can regulate the gene expression at transcription level. So at transcription level I can regulate. So this means if I if I don't want that gene product, I can stop that process of transcription first, right? So that is what I mentioned at transcription level. So this will be, that is the formation of primary transcript. So the primary transcript, as I told, in case of eukaryotes, the DNA will first give rise to hnRNA. So this is what I call primary transcript, and that later undergoes the processing like RNA splicing, 5 and capping, third and tailing, and all, and that finally results in the mature, okay, mRNA formation, right? So at transcription level, so that will be, right, uh, in formation of primary transcript. So at this level, okay, the cell has mechanism. So it at this level itself, so it can stop the expression, the gene expression. Okay, so that is at transcription level. Then, right, next one, processing level. So processing level, so I told you what we mean by processing. So that will be, right, the regulation of splicing. If splicing does not take place, or if we stop that splicing, then the mRNA will not form. If that mature mRNA doesn't form, there will be no process of translation. So that is what the regulation we can do it, okay, or the cell itself can go with the processing level. So that regulation of splicing. Then at another level is transport of mRNA from nucleus into cytoplasm. So once the processing and everything is completed, the mRNA need to come out of the nucleus for the process of translation to take place. So I told you in the earlier sessions that the formation of RNA processing and everything that will take place inside the nucleus in eukaryotes. Once that takes place, the mRNA will be transported out of the nucleus into cytoplasm. So this is nucleus and this is cytoplasm of the cell. So what I mean that transcription will take place in the nucleus, whereas translation will take place in the cytoplasm. That is in case of an eukaryotic cell. But in case of prokaryotic cells, there is no such compartmentalization. So there is no nucleus. So hence what happens, the transcription and translation can take place at the same site. Right. So if I stop this mRNA, right, or the cell itself does it, then the mRNA will not be coming into cytoplasm and there won't be translation taking place. So that is okay, we can regulate the gene expression, right, by stopping the transport of mRNA from nucleus into cytoplasm. Now at the last level, that is translation. So translation level. Now the eukaryotic cell has the ability to stop the gene production at 4 levels. So it can stop at transcription level, it can stop at processing level, it can stop at transport of mRNA from nucleus into cytoplasm, and it can also stop at translation level, right? But in case of prokaryotes, it's very much limited. So in prokaryotes, right? So the gene expression, so the regulation of gene expression predominantly is at transcription level, right? So in case of prokaryotes, the regulation of gene expression predominantly is at transcription level, right? So that's about gene expression, how it can be regulated in eukaryotes, at what levels it can be regulated in eukaryotes and in prokaryotes, right? So now let's come back to this, the Lac Operon. So what we mean by operon? So operon is a polycistronic structural gene, a polycistronic structural gene, right, that is regulated by a common promoter and operator. So that is regulated by a common, right, promoter and I'm sorry, promoter and a regulator. polycistronic structural gene that is regulated by a common promoter and a regulator. So that's of course I'll say regulator gene. So promoter gene and regulator gene is going to regulate a polycistronic structural gene, and we know this polycistronic structural gene will be seen in case of right prokaryotes. So I'm going to discuss this Lac Operon in E. coli. So in E. coli, right? So as I already mentioned, when we put E. coli into the lactose medium, it's going to produce enzymes that will metabolize the lactose. And when we remove it from the lactose medium, it stops synthesis of those enzymes. So how the bacteria comes to know that when to produce and when not to produce, that is what the regulation of gene expression, and we will understand it by the Lac Operon. Right now, when we see in the E. coli DNA molecule, so in the E. coli DNA molecule, so here the P stands for promoter region, okay, and then the I stands for inhibitor. So should not get confused with the inducer. Later I am going to tell you inducer. Inducer is different and inhibitor is different. So here we are going to see genes in this order. So this is a promoter gene and this is a inhibitor gene. So this is DNA molecule on which the genes, we know that they are present, okay, in a linear fashion, and this is the promoter, and this will have a sequence of nitrogen bases. The inhibitor will also have sequence of nitrogen bases, okay, and after the inhibitor we have again promoter, right? So promoter, then after that we have operator. So here O stands for operator gene, right? So all of them, whatever I'm mentioning, we know DNA what will be having inside it. So it will be having a sequence of nitrogen bases. That's it. So here this is the region of DNA which will be having a particular sequence of nitrogen bases, which I called here it as promoter, and then the inhibitor. So this is the order that we need to follow. We cannot change this order, okay, because the genes are present on the DNA in this order only. Then after this promoter we have actually three structural genes, that will be Z, Y, A, right? So these three are the structural genes, right? So structural genes. Now what these structural genes they code for? So here the Z gene, the Z gene, it's going to code for beta galacticide. So it's going to code for, sorry, galactosidase is enzyme. So it's going to code for beta galactosidase is the enzyme, right? You can write in short with beta gal. So that's enzyme. So all these structural genes are going to code for enzymes. So this is enzyme. I hope you all know that whenever the name ends with the ASE, it's an enzyme, right? So need not every time tell that this is an enzyme. So whenever the word ends with the ASE, so that is an enzyme. So Z gene will code for beta galactosidase, and the next one, the gene Y, will code for, that is gene Y code for an enzyme called permease, an enzyme called permease. And then the A gene, this codes for an enzyme called trans-acetylase. It codes for, okay, trans-acetylase. Now all these three enzymes are required for metabolizing lactose. Okay, so metabolizing lactose, and now the production of these genes will all depend on the presence of the lactose, right, or absence of the lactose. In the presence of the lactose, all these three genes will be expressed, and in the absence of the lactose, all the three genes will be stopped, means they will become inactive, or you can say they will get switched off in the absence of lactose, right? So of course we can say that the expression of the gene can depend on metabolic, physiological, or environmental conditions that actually regulate the expression of genes. So in this case, uh, right, it is the environment conditions that regulate the expression of gene, whether it is present in the lactose medium or out of the lactose medium, right? So these are the sequence of genes that are present on the DNA of E. coli. So DNA of E. coli bacterium. Now, so here the inhibitor will be transcribed. So this will be actually called the regulator gene. So inhibitor is a regulator gene. So this is the one which is going to control the expression of these structural genes, right? So when this regulator gene is transcribed, it's going to produce mRNA, and this mRNA, when translated, is going to produce something called the repressor protein. It's going to produce repressor protein, and this repressor protein has affinity to the operator. So it has affinity to operator. So this one, the repressor protein, will go and bind to operator. So you can see here the repressor protein is bound to operator, right? I can say here genes are constitutive or inducible. So the genes can be constitutive or inducible. So this gene, that is regulator gene, will be expressed all the time. So hence such genes are called constitutive genes. So regulator gene is expressed all the time. So it is expressed all the time. So hence the regulator gene is a constitutive gene. So this is a constitutive gene. So it's a constitutive gene, right? So hence in small, okay, amounts, the repressor protein will be continuously formed, and that repressor protein, okay, so will go and bind to operator. Now we know that for the process of right, transcription, so earlier we have studied the process of transcription, and the RNA polymerase will come and bind at the promoter. Yes, So here this is the RNA polymerase enzyme. So in short I write RNAP. So here RNAP stands for RNA polymerase enzyme. Stands for RNA polymerase enzyme. We know that the RNA polymerase will bind to the promoter, but here as there is a repressor protein bound to operator, the RNA polymerase cannot move forward to transcribe these structural genes. So this is what I say in switched off condition, means these structural genes are switched off. Okay, and the genes that can be switched on and switched off are actually called inducible genes. So here these structural genes are actually inducible genes. So they are inducible genes because they can be switched on and switched off, whereas this one, the regulator gene, is not the case. So it will be continuously expressed. So here the structural genes expression can be controlled, so hence I say they are inducible genes. When the E. coli bacterium is out of the medium, out of the lactose medium, there is no need, okay, for these structural genes to get expressed. So hence now they are in switched off condition, and the switched off condition happens when the repressor protein binds to operator and doesn't allow the RNA polymerase to transcribe the structural genes, right? So this is in the absence of inducer. So in absence of inducer. So what is that inducer? So the inducer is either lactose or allolactose. So inducer is lactose or allolactose. If this is not present, then this is the condition what happens, and now what I say, it is right switched off, because unnecessarily producing the products of these genes is a waste of energy. Hence now the structural genes are in switched off condition in the absence of inducer, means the lactose is not present, right? Now when you put this E. coli in the lactose medium, so when we put this one into the lactose medium, so let's see, in presence of inducer. Okay, so now when the inducer enters, okay, so here lactose molecules enter the cell. Okay, so these are all the lactose molecules, lactose or allolactose. So lactose or allolactose, and this is inducer. So this is inducer. So when it enters into the cell, the lactose molecule is what it's going to do. It goes and binds with this repressor protein present on the operator region. So it's going to come and bind over here. So it comes and binds to this, repressor protein that is present on the operator, and this results in the formation of inducer repressor complex. Inducer repressor complex, right? Okay, of course, we need to keep in mind the expression of this. Okay, so that will be the expression of Lac Operon will be present at its minimum level all the time, and if it is not the case, then lactose will not be able to enter the cell. So here, once the lactose molecules enter the cell, they are going to some of these lactose molecules. So say here one lactose molecule, it bound with the repressor protein that is attached to operator, and it results in the formation of inducer repressor complex. So when inducer binds to the repressor protein, there will be a structural change in the repressor protein, and this results in unbinding of this repressor, inducer repressor. This whole complex will be removed out from the operator. So hence this whole thing is removed. So now when there is no repressor protein on the operator, so the RNA polymerase have no obstacle. It has no obstacle. Now it will be moving to the structural genes, and that results in the transcription, and that will finally result in the formation of a polycystronic gene, because this will be having information for formation of three enzymes. Okay, so this will be the one with Z, Y and A, and when translated, it's going to produce beta galactosidase, permease and trans-acetylase. Okay, so this is how we see, okay, when E. coli is put into lactose medium, so in the presence of inducer. And I told you that the repressor protein will be continuously produced. So it's a the regulator gene is a constitutive gene. Okay, now this process will be continuously. So next repressor protein, when it is formed, one more lactose will bind to it. Okay, it will bind, and it will not allow to bind it to the operator as long as these lactose molecules are in sufficient concentration, right? So once that decreases, then what happens, the repressor protein will not have if it is not bound with the lactose, then the repressor protein again goes and binds to operator. So you have to keep in mind, every time the transcription process need to take place, the RNA polymerase have to bind with the promoter and move up to this A. Once it is finished, it need to unbind and again come and start with the promoter region. Now this time, when it started with the promoter, we have here the repressor protein. So hence now the expression of these structural genes is stopped. Right. So this is how we see the expression of these structural genes, which are called inducible genes. Okay, so now I say this is switched on condition. So it's a switched on condition. Now let's see what does this beta galactosidase function, what it does, the permease and trans-acetylase, if they are produced, and how they are going to metabolize this lactose or allolactose. Right. So here the lactose will be acted on by the beta gal. It's going to convert it into galactose and glucose. Okay, and permease its function is, okay, it binds on membrane and increases permeability of lactose into the cell. So that is the function of permease. Then coming to trans-acetylase, right? So this trans-acetylase, so whose function is to, okay, transfer, okay, so it's going to transfer a acetyl group, going to transfer acetyl group from acetyl CoA to, right, the molecules like lactosides to lactosides, okay, or galactosides, etc. So that is the function of trans-acetylase. So transfer acetyl group from acetyl CoA to lactosides or galactosides. So that's the function of all these three. So lactose, permease, and then the third one, trans-acetylase. Right. Now, lastly, the regulation of Lac Operon by repressor. So here, the regulation of Lac Operon by repressor is what we call it a negative regulation. Is called negative regulation. Okay, so whatever we have studied is negative regulation. Okay, of course, in case of positive regulation, it will be opposite. Okay, that we will discuss it later. Regulation of Lac Operon by repressor is what we call negative regulation. Right, so that's all about the regulation of gene expression. Right, so in the next session we'll see about human genome project.